Good evening. My name is Misha Stone, and I'm a Reader Services Librarian here at the Central Library. Thank you for coming to the Central Library tonight. It's good to see you all here. We are excited to be hosting this event tonight with the defeatHIV community, the University of Washington Center for AIDS Research, Fred Hutchinson Cancer Research Center, and the UW AIDS Clinical Trials Unit. It is an honor to welcome back native Seattleite Timothy Ray Brown, a.k.a. the Berlin patient, and the doctor who cured him, Gero Hütter. I want to say a special thank you to the Seattle Public Library Foundation, who makes so many of our free library programs possible. And I want to mention that this event is being recorded for podcast. It's always a good reminder to turn off your cell phones at this point now. Thank you. You can subscribe to the library's podcasts at www.spl.org. And now I'm going to turn the evening over to our lovely guest speakers, and first, Michael Luella.
And before Misha goes, I just wanted to say thank you to the library and thank you to Misha. And there's actually a card here for Jared, who also helped us. And hold on, I have a present in my pocket here. Oh, wait, wrong pocket. Sorry. Here you go. USB drives with the CURE classes that we taught on Saturday for you. Yeah, I guess that was German. So welcome, everybody. I'm glad to see you all here. Tonight is your night, and you get to really drive the conversation. We have brought here the man who has been cured of HIV. He was here 2 years ago, but you wouldn't believe it. Some people have not heard. In fact, on our Facebook page this time around, I got a lot of sort of snarky comments of like, well, I'll believe it once I see it in the world headlines. And true. So it's amazing. I think events like this are important to do because still, no matter what, no matter if you're on the front page, top of the fold of the Seattle Times, no matter if you're on the news, on TV, it's not getting out there that something has changed. And so you're going to be able to hear about that tonight and think of this as your night in the sense that you will be able to ask the man who has been cured and the doctor who thought up of that experiment that led to it and that has led to a whole field of science called HIV cure research. We have now scientists all around the world working on ways that we might be able to either cure it, meaning HIV, or maybe make it like cancer, go into remission where people will not have to take a daily pill every day for the rest of their lives. So it's exciting to have that science, and you're going to be able to ask your questions about that. But before we get to that, I wanted to just sort of remind you of some things. First of all, she told you about your cell phones being turned off. But, you know, don't really turn them off, just turn them down so there's no sound, because one of the things that I like to do when I go to events is take pictures so I have lasting memories. And so if you all would love to, I would love for you to take a picture right now or as they come out or anything like that. Feel free. And if you want to put it on Facebook, you can always hashtag it with #defeatHIV, all lowercase and all together. And it would be great to see what pictures that you snap of people here tonight doing that. And so that's what I would love to see. Your cell phones could be out just silent. And watch, I'm going to take a picture of you all right now. Oops, watch out. Ready. Smile. Oh, wait, kimchi. It's not working. Kimchi, cure, yes, cure, definitely. Look at that. All right, and got Chanel in there too. Thank you. So I'm going to introduce our moderator, who's one of our community members, a part of our community advisory board for defeatHIV, and his name is Mullen. And we also have other community advisory board members here, and you might notice them because they have these clipboards that they're carrying. What they're really trying to do is bolster our email list. I send out emails about events like this, about our monthly CAB meetings, and as well as any sort of latest HIV cure research news. So if you would like to get that into one of your email inboxes, you could print your name and print your email address. At some time during this event, we'll have those clipboards the entire time. Feel free to write it down, and if you would write it really neatly so I don't have to try to guess if it's an M or an N, that would be great, and that would help us a lot. So I'm going to introduce Paris, who's going to introduce our two guests. So thank you very much for coming, and enjoy the event. Here you go.
You can do better than that. Clap. It is an honor to be here tonight with you all, and to be specifically with the two folks I have the evening tonight, and specifically Timothy Ray Brown. But before we go on, I kind of like to warm up the crowd and just kind of loosen things up. So if you will do this just for me, turn to your neighbor and tell them, say, hey, you have never looked as good as you do right now. Would you just tell them? You've never looked as good as you do right now. Don't talk to them about how they looked last week, but just talk to them about right now. You've never looked as good as you do right now. And I mean it. I've never looked and it only gets better. And if you didn't mean it, just keep looking forward, and they won't even get it. They won't even get it. So as you know tonight, we're here with Gero Hütter and Timothy Ray Brown, and thinking about being with you tonight and moderating this time. One thing that I thought was really important to me was to really highlight and bring forward the humanity of the people that are involved. Not just a patient who underwent a procedure and now is cured, which is amazing. Not this doctor, physician guy from Berlin that did this amazing thing, but to really bring forth the humanity of those involved. And I'd like to start from the perspective of those human beings, those individuals, mothers, daughters, husbands, wives, sons, children, people, all over the globe, for many years, decades, many years, who have willingly participated in research or who have given their bodies over to certain medical procedures, being uncertain of the outcome, not sure if they would live. All this with the hope of advancing our science, advancing research, to develop better medications so that people can live longer and healthier lives. And so these were people who sacrificed so that we could be here today. And so if we could just, a moment of silence, just kind of honor those, many of those who will never know their names at all, who participate in clinical trial research, whose blood is drawn, who's poked and prodded all over the world to help advance science and make it possible for us to be here today. A lot of the communities, if I can get more explicit with you, for talking about HIV, a lot of the communities that often faces a lot of discrimination and face the disparity. We're looking at homosexual people, many of those in the LGBT community, for if it wasn't for those MSM, that's what gay men are referred to, men who have sex with men, when we're talking about clinical trial research, MSM and transgender persons who had given up their lives, time their lives, their resources to participate in trial research, we wouldn't be here. We would not be here. And that's just not just for HIV, but that's for so many other modalities and interventions that we have today. So in honor of those human beings, a moment of silence, I would ask. Thank you. Thank you. Okay, so now, we don't have to be so somber now, because I still look good and I'm easy on the eyes. So that should make it exciting for you, okay? I want to introduce you, so thank you, but we're excited. This is a joyous time. I mean, look, we are in 2015 talking about cure of HIV, a virus that unfortunately has claimed the lives of too many people. An infection that still, unfortunately, people are contracting still today. 6,000 per day globally. And we are living in a day and age where no one has to contract the virus anymore. And now we have someone who has been cured of the virus. So what I will do is introduce, with no further ado, Timothy Ray Brown and Gero Hütter. But I'd like to read their brief bio, if you'll indulge me for a moment. And I could just say, hey, Tim, come on up, Tim, but I want to do him justice in kind of reading some things that you may not even know about him. Timothy Ray Brown is editor of the CURE Report and co-founder of the Cure for AIDS Coalition and co-founder of the World AIDS Institute. Dubbed by the media the Berlin patient, he is the first person in the world to be cured of HIV. Timothy's story, arguably one of the most fascinating and followed in the realm of HIV research, began in 1995 when he was diagnosed with HIV while attending school in Berlin. For the next 11 years, doctors treated him successfully with antiretroviral therapy. In 2006, however, Timothy was diagnosed with acute myeloid leukemia and underwent chemotherapy. While the first round of treatment appeared to work, it also made him more susceptible to infections. Brown developed pneumonia early on in his treatment, and he battled sepsis halfway through his third round of chemo. His doctors realized they would have to try a different approach. His oncologist, Dr. Gero Hütter of the Charité Hospital in Berlin, opted to give Brown a stem cell transplant to treat his leukemia. And after that, it is history. He's cured of HIV, and we're going to hear more about that here in a moment. Dr. Gero Hütter, MD, is a chief scientific advisor of the CURE Report and Cure for AIDS Coalition. He is also head of the stem cell unit at the Institute of Transfusion Medicine and Immunology Mayhem of Heidelberg University in Germany. Gero is a German hematologist. Hütter's and his medical team transplanted bone marrow deficient in a key HIV receptor to leukemia patient Timothy Ray Brown, the Berlin patient, who was also infected with HIV. Subsequently, the patient's circulating HIV dropped to undetectable. The case was widely reported globally in the media, and Hütter was named one of the Berliners of the Year for 2008 by the Berliner. Now, this is some of this is in German, and I'm not German. There are black Germans. I'm not one of them. I just want to be clear, okay? In 2009, Hütter, Eckhart Thiel, and others from the Charité Hospital in Berlin, Germany, published a report on the case in the New England Journal of Medicine. Their patient, Timothy Ray Brown, a U.S. citizen from, drumroll please, Seattle, Washington, and living in Berlin, had both acute myelogenous leukemia and HIV. The physicians found a bone marrow donor with a CCR5 Delta 32 mutation in both genomic copies of a gene encoding of a cell surface. Blah, blah, blah. It goes on to say more. I'm going to stop. So I'll tell you what that means. Basically, there is, and we'll learn about this here today, that there's a percentage of people in the world who have what's called a CCR5 or CCR5 Delta 32 mutation, which means that essentially it's impossible for them to contract the virus. And so they had to find a donor for Timothy Ray Brown who had this deletion in order to do this bone marrow transplant. And it was successful. The probability was very low that it would be successful, and we'll talk about that probability here today. So enough from me. I would love for you to please stand to your feet and applaud Timothy Ray Brown and Dr. Gero Hütter.
And before we get started, one of the things that you didn't know probably is the date that this happened. Does anyone know the day that Timothy got his transplant? December 1st? No? Pretty good guess. Uh-oh, there's one person, his mother. Now, we're talking about his first transplant. Do you remember the date? It was February. Woo! You got it. We're going to keep you right there. So, there's something that happens in transplant culture that when people get a transplant, they refer to that day as their birthday. And you may have heard the similar things. Some people recognize their HIV diagnosis day as a new birthday because a new life from that moment on. And so we are actually going to celebrate February 7th, 2007, which was the date of his first transplant and the date, you could say, that cure of HIV was born. and it's an important date for us all to remember and memorialize, February 7th, 2007. And so we have a cake here, and we're going to sing happy birthday to Timothy. So why don't you bring on the cake? And you'll see there's a little 8th candle there, and it says on top of it, 8 years cured, and February 7th, 2007. So please, let's all start singing a 1, a 2, a 3. Happy birthday to you. Happy birthday to you. Happy birthday, dear Timothy and the Cure. Happy birthday to you. Why don't you bring it over to him to blow out? Thank you. Yay. Whoop, yeah. And so, while we get proceedings, we already have some pre-sliced slices of cake, and we'll have people handing it out. So if you want some cake, it'll be coming to you, okay? So enough from me, and here you go, Paris. Back to you.
Wonderful. So, to let you know what we're going to do tonight, we're going to hear Timothy Ray Brown's story, obviously from his perspective, and also to let you know, this is the first time that Gero and Timothy have actually, or one of the first times, have actually been in a symposium or forum together. They have been interviewed around the world separately about the experience, but one of the first times that they've actually been in the room together talking about this amazing feat that has happened. So you'll hear about it from both of their perspectives. Towards the end, you'll have an opportunity to ask your own questions. So be thinking about what you'd like to ask, because you get an opportunity to do that at the end. So I think with no further ado, we'll begin. And Timothy, would like to start with you and kind of bring us back to the time before, well, as I was reading your bio, I thought it was interesting. I mean, the time maybe perhaps when you came to Berlin, you contract the virus, and kind of give us some history before these amazing events happened in your life.
Okay. I have a mic on. Yeah, okay. Can everybody hear me? So I took a trip with 2 female friends of mine around Europe, mostly Western Europe, but we also visited East Berlin and Greece. So that's not West Berlin, really, but it is now. Or no, not Berlin, Western Europe. So anyway, I got back to Seattle, and I was working in banks at the time, and I decided I was bored with that, and I wanted to move to Europe. And actually, Barcelona was my first choice, and I had been to Berlin, and I liked Berlin, but Berlin was kind of a mess back then, and so it was in transition because it was right after the wall came down. And so I moved to Barcelona, and then I took a trip up to Berlin to meet my friend Tony, who's here, back there, and a couple of other people. And this was back in the days without cell phones. And so we never got hold of each other. So I ended up spending the whole month in Berlin, but that was okay. I met a lot of people and had fun. And so I went back to Barcelona and got kicked out of my apartment because I had a very homophobic roommate, a Syrian guy. Nothing against Syrian people from Syria, but he was kind of a dick.
That's going to be edited out, don't worry.
So anyway, I was, my bedroom was actually a closet in the apartment, and it was like 90 degrees Fahrenheit and very warm. And so we slept out in the living room on the floor. And then the Syrian guy got up in the morning and said, "Why are you here?" He made his coffee, and then he said, "You were out here naked on the floor because the covers came off and we were naked, and so you do that again, I'll throw your stuff off the balcony." And so I thought, okay, throw my stuff off the balcony, your stuff's going off the balcony. and then that happened again the next night, and that time he said, "You do that again, I'm throwing you off the balcony." And I thought, okay, this is getting dangerous. And so I talked to the friend who was visiting me from Berlin, and he said, well, you can come and live with me in Berlin. So that's how I ended up in Berlin the first time. Matthias was his name, and that's his actual name because he gave me permission to use it. So anyway, I went back up to Berlin. I hitchhiked, and it was kind of dangerous, but that's okay. I got through it. So I got up to Berlin and started living with Matthias. We broke up for some reason, and then I ended up meeting somebody else, and he wanted to move back. He wanted to move somewhere in some European big city in Europe. And so we picked Madrid. And so I went ahead because he still had to work in Berlin. And so I ended up going back to Barcelona just to stop there. And anyway, I saw old friends that I had met in Barcelona. And I got two apartments rooms in two apartments for both of us. And so we ended up moving back to Barcelona. And I was there for about a year and a month. And then I decided I wanted to study more. So I didn't want to learn Catalan, so I decided to move back to Berlin. I had just learned Spanish, so I didn't really want to learn German. But I moved back and started. I started taking classes, and at some point I had sex with somebody, and I used to use the withdrawal method, and it didn't work. He didn't understand what I was saying in Spanish, and so I went to Roosevelt. That was the first high school I went to. Then I went to Ingram. I like Roosevelt better. So I ended up getting infected with HIV, I guess, and the partner that I moved back to Barcelona with didn't want to get tested because he didn't want to know if he was positive or not. Because at that time, HIV was a death sentence.
What year was that?
That was 1992 or something like that. Yeah, and so I didn't get tested. I had gotten tested in Seattle in 1990 was the last time, and then I didn't get tested in Europe at all until in '95. A good friend of mine and former partner told me that he had tested positive for HIV. And we had not used condoms and not had such sex. And so I went and got tested thinking, okay, I may be positive, I may not be. And sure enough, I tested positive for HIV.
What was that like, learning of that diagnosis for you?
It was still a death sentence because there was only AZT at the time. And the doctor at the Berlin clinic called the Topin Institute, which is the Center for Tropical Medicine. They do work around Africa and wherever. So it was a death sentence for me. In fact, that friend who had told me that he had tested positive said, we only have about 2 more years to live.
2 years, that's what I read, and that was 1992, you said?
95.
95.
Yeah.
And so then there was an advent with regard to antiretroviral therapy.
Yeah.
And that was a game changer.
Exactly, it was.
For many.
So I took small doses of AZT for a year, and then protease inhibitors hit the market. And so I was able to take one, and then that got switched to a triple combination, and in '96, and then I basically lived a normal life, and I thought that I might live for a normal lifespan.
So it was about 10 years, as I read, from the time that you were diagnosed with HIV to the time that you learned of the leukemia. Is that correct?
Yes, correct.
So in comes, and I understand the story, you, well, why don't you tell us about that? I understand you're feeling weak.
Yeah, I had taken a trip to go to a wedding in New York City, and I was tired the whole time, and I didn't know what was up. I thought it was jet lag, but then I went back to Berlin, and I used to ride my bicycle about 10 miles to work, and it took me longer than usual.
10 miles to work.
Yeah, and back. So I was in shape.
Okay, that's amazing.
Yeah, I was in shape.
You were in shape.
Yeah.
I hope so. Yeah.
And anyway, I went about a mile to go to lunch, and I had to stop halfway along the pathway, and I called my boyfriend at the time and said, I'm not feeling very well, and I had problems getting to work, and my boss chewed me out, and I think you need, can you call my doctor? And so he called my doctor, and I couldn't get an appointment for the next day, so I ended up going to his HIV doctor, HIV specialist. And so I went there the next day, and they checked my blood and found out that my red blood cell count was really low, like almost 0. And so I needed to get a transfusion of 2 liters of blood. And so I got that, and the value would go up, and then the next day it was down again. And so that continued for about a period of a week, and then he finally sent me to an oncologist.
And this is where you met Gero.
Yeah, I went to the oncologist. He said, my gut feeling tells me it's nothing serious, but then he did a test that showed that I probably had something.
And Gero, you were the oncologist
Yeah.
there. Can you tell us about the time when you met Timothy in this condition?
He was in a quite good condition at this time. He came to us, and he was weak, that's right, but he was not too bad. And we make a bone marrow punction for our diagnosis, and we saw that the same day when he came the first time that he has leukemia. And it took another day, and we started therapy.
Started therapy there.
Yeah.
Okay, and then so where did you get, so we're taking care of leukemia now to keep him alive. When did the thought of addressing the HIV come into the picture? How did that conversation start? Why did you think about it? And you know, as we discussed earlier, I kind of want to get an idea, maybe we can give the audience a sense of where did you get this idea that, hey, if we can take care of this leukemia, let's address the HIV, something that had not been tried to cure, eradicate, something that had not been done before. So tell us, what were you thinking?
Yeah, at this time...
Gero, what were you thinking?
My knowledge about HIV at this time was very limited. So
Sure.
I'm an oncologist, and we had not much to deal with HIV patients. It was not our typical patient. But I read an article 10 years ago when I was a medical student where this mutation was described which confers natural resistance against HIV transmission. It's this CCR5 delta 32 deletion. It means that these people who have this mutation don't express CCR5 on the cells, and HIV cannot enter the target cells. And this was so impressive for me as a student that there's a natural resistance against HIV that I kept this information for 10 years in my mind. And at the moment where Timothy went to the door, and I saw that he has leukemia and he has no siblings, so we need an unrelated donor, there was clear for me that we have to find a donor who has this mutation.
Okay, so here it is. I want to frame this discussion as we move on this piece, talking about the laws of probability. Now, Timothy shouldn't be here right now, okay? Was it impossible for this procedure to be successful? No, but the probability was extremely low. So the first thing that was kind of improbable, perhaps unlikely, we have a doctor who 10 years ago had read about the possibility of being able to cure HIV with regard to a transplant as it pertains to the CCR5 delta 32 deletion. Keeps that filed away in the back of his mind. 10 years later, you're the one that this call was directed to meet Timothy Ray Brown. And here you recall that information, the study that you had read, and thought perhaps that it could apply to this. So I'd like for you to talk to us about the likelihood or the lack thereof with regard to him even coming through and then coming out alive. If you could tell us more about the CCR5 delta 32 deletion, how many people in the world is that true for that possess that, that have that mutation?
Yeah. It's around, in Northern Europe, it's around 1%.
Around 1% in the world, that's it.
So to make it clear for you here in the room, probably 100, 150 people, and there are at least mathematically one here sitting there with the mutation, and another one here in the audience. So
originally it was just half of it. Heterozygous. I don't know for which parent.
So it's not uncommon. It's a thing. What happens? 1%. It's not much. On the other side, the probability that Timothy and I were matching in tissue typing for allogeneic transplantation, it's far below 1 to 100,000 or 200,000, probably 1 to 1 million. So to find here in the room someone who is matching in a tissue type with any of us is quite uncommon. And this is the trick is that you're increasing the number of registered donors. And there are now 20 million donors for stem cell transplantation registered worldwide. So if you're increasing the number, the probability increases that you have matching donors. And in Timothy's case, it was 232 donors who were matching on the tissue type, and 1% from 232 are 2 who are homozygous. So you have to test these all. And then at last, we found one that was matching with the tissue type and the CCR5 deletion. So.
There we have.
Yeah.
Okay, and if you talk about the healthcare system, why this probably would not have happened if this had taken place in the United States. So good thing that you were in Berlin. Can you tell us about that?
Definitely.
First, I have to say that the principal idea in doing this approach is not mine. So there were many people before who are oncologists and infectiologists who are aware of this possibility, and they worked at this possibility. And in 2001, here in the US, a big stem cell corporate unit was built, and all of the corporate units were tested for the deletion in 2001. So six years before we started all this in Berlin. So they were aware of this possibility and the work, but it's not too easy to use cord blood. And the other point is that for unrelated donors, in the US, we have very strict restriction in finding or getting access to material from the donors. And this is another situation in Germany. We have a very close contact to our registries, and we ask them for doing this. It's not usual to do this, but they agreed. They saw the possibility to help us in this point, and so
there was a registry.
Yeah.
At that point.
At that point, the registry, when at this point the registry said no, we want to do this, the whole project was cancelled. So.
And we still don't have one in the U.S. So
that's right.
That's something that needs to change. So then the procedure. If you could talk about the procedure then, and what did that look like, and the rationale with regard to why it was going to work for.
Yeah, Timothy, we started with the regular chemotherapy, like all the leukemia patients have. And at some special time point, we have to decide how to go on. Do we stop there or do we make this transplant? And when we making this transplant, he needs something to get rid of the old immune system, or to get rid of the bone marrow. It's usually a combination of chemotherapy and irradiation, and then the stem cells from the donor were reinfused like a blood transfusion, and 2 weeks after this, the immune system is completely exchanged, and the patient can leave the hospital.
And what's the likelihood for one surviving that procedure?
Yeah. It depends on the age of the patient and the diagnosis. If you have something like aplastic anemia, this is not a cancer, it's an autoimmune disease, you have up to 99% survival. If you are 65, have difficult leukemia, the survival rate drops down to 50% or less. So it's, in his case, I think the good estimation for the survival would be 80%. So
Okay.
a good chance to survive this.
Timothy, so we know that first it was leukemia and HIV was secondary.
Right.
And so it's kind of, let's see what happens. So Gero's moving forward in faith.
Mm-hmm.
Obviously, had discussions with regard to what would be the implications of going through this procedure.
Right.
What was your thought process with regard to deciding to move forward?
At the time, I talked to friends and family. I talked to my grandmother, and I called her on the phone, and I said, well, okay, I'll survive with HIV, but my main goal is to get rid of this leukemia, because I can die very quickly from it.
Right.
And so at that point, my leukemia, sorry. At that
Take
point,
your time and have some water.
at that point, my leukemia was in remission. So I actually said no to the transplant because I thought, well, I can go on taking HIV medication. My viral load was at 0 or undetectable, and I was doing quite well with that. It was the leukemia that was the problem, and so I said no, and then the leukemia came back. It went out of remission, and at that point it became clear that I had to get the transplant to survive.
How many procedures, all in all, after did you have to undergo?
3 normal chemotherapies
Yeah.
and 2 transplantations.
Right.
And 2 transplantations.
Yeah.
Okay. I had asked you before if you had the opportunity to do this again, Gero, would you?
Yes, but not in Timothy's case, because
Yes.
he has
Because they don't get along.
Not at all.
That's not the reason.
No, it's unusual to do a second transplant. You're increasing the toxicity in every transplant you're doing, and 3 transplantation is, I think, much more than you can have as a patient. So.
He actually told me, this was at Washington University in St. Louis a couple years later. We asked him, what do you think my survival chances were? And he said 5%. I'm like, oh wow, I'm glad I didn't know that at the time. I probably would have given up then. So yeah, I'm glad you didn't tell me that.
Gero, you left that little piece of information out there. You had said today when we were talking what you believe, because we had the conversation with regard to the probability of science. We know the numbers there with regard to survival and the chance of things becoming successful. And there is this other piece of, you said, luck. And did you want to kind of tell us what are some of the lucky factors that happen in this case here? Because I think some folks will be wondering, we'll address this as, why can't this happen to everyone else? We will talk about what we call the Boston patients and how a similar type of procedure has taken place here in this country, however unsuccessful. But, why can't this happen with everyone else? Or has it?
It has happened. We have a patient, you met him, this boy who received a cord blood unit with a CCR5 deletion, but unfortunately the patient died from side effects of the transplantation. And there is another 5 patients who received such a special transplant, but none of them survived very long, so we have no follow-up data. And we cannot be sure what happened with the virus. They were all on therapy during the transplantation and afterwards. So after all, Timothy stands alone still.
I want more people to join my club.
but principally, it's not a limitation, a medical limitation. It's just a question of possibilities you have. And if you are looking in I think in all the world, 10, 20, 30 patients with HIV receive allogeneic transplantation. No one knows the exact number. And if you are looking for everyone very closely for a matching donor who has this deletion, I guess that 1/3, 20%, would have the possibility to receive such a transplant, and we have much more data to make final conclusion. Well, how successful is this treatment for these special patients? It's always patients with leukemia or lymphoma or some kind of cancer. It's not the normal HIV patient without concomitant disease. So.
Amazing. So you said that Timothy had this desire or passion or will to live, which you believe was very instrumental in the whole process.
That's right, yeah.
So how did your life change after you had been cured? How did your life change? So physically, if you can talk to us about some of the rehabilitation or therapy that you are having to go through or have gone through after the procedure.
Okay, I was sent to this center for people with severe brain injuries in Berlin, and at that point they rolled me on a bed, and one of the physical therapists came to my bed and said, "Why lift your right leg?" And I couldn't tell the difference anymore. I lifted my left leg, and so I started out really in bad shape. And then they got me into a wheelchair, so I rolled around in the wheelchair and hit walls and stuff like that. And then another physical therapist got me up on a walker, and so I could walk with a walker. And then I graduated from that to being able to walk without the walker. And it was tentative. It was like a baby learning to walk. Yeah.
All over again.
All over again, yeah.
Something like that.
And a good friend of mine from Seattle, who now lives in D.C., came to visit me, and she said that she told another friend of ours that she came to say her goodbyes to me. And she got there, and I was up and walking. And then, yeah, and so I got up out of the hospital and went to a rehabilitation center, and I would walk there to a restaurant nearby, and I didn't like the food at any hospital. So I would go to a restaurant, and so I was able to walk to the restaurant, and then I tried walking up a hill and slipped down and fell. Yeah, but so I got to the point where I could kind of walk semi-normally, and I still have limitations in my walking ability. I go to the gym like 3 or 4 times a week now, and I always do the treadmill, and I can go up to like 15 miles an hour, and so I can run pretty fast. I can't run on pavement, just treadmill.
Okay. Some folks don't even do that, so that's good. You used to bike to work 10 miles. Now you're in the gym 3 to 4 times a week. That's another session. Good for you. We can learn from that. So, Gero, have you tried to attempt to do this procedure to anyone? The bone marrow transplant, the stem cell. Have you attempted this again, or?
No, I have requests after we published the report from Timothy. We had requests from all over the world.
Sure.
And I worked on 22 requests from other institutions, and we found 4 matching donors for them. And in one case, this transplantation happened, but it was another case where the patient died rapidly after the transplantation, and the other 3 that were transferred before the transplantation was done. Yeah, that's a problem that we have not so that access to people who are matching for this kind of study, and we're trying to expand this information to more countries, to more facilities, to more treatment units, and to get more patients for that.
Absolutely.
I've got a question for you.
Yeah.
Are you asked to be an advisor on any of the projects that are going on in Europe?
In Europe,
No.
yeah. Well, no, not as an advisor.
okay,
We're
okay.
part of the project,
Oh,
yes.
good,
Yeah.
good.
Now, Timothy's cured. Can you tell us what exactly that means?
Yeah, it means that his immune system was exchanged and replaced by cells who have the natural immunity against HIV transmission. And we have to say that after the transplantation, there must be many infected cells, residual cells after transplantation procedure, and they can reemerge. We learned it from this Boston patients who received normal CCR5 normal cells and relapsed from HIV and rebounded after several months after the treatment was stopped. So the lesson from this is that probably just one infected cell is enough that HIV can respread the immune system if it's not protected. And this is a special condition in Timothy. His immune system is protected. We know it from these who have this mutation naturally, that the probability in getting infected is very, very low. It's not 0, but it's very, very low.
Very improbable, like me marrying Kim Kardashian.
Yeah.
That's
right.
okay.
And I think this is when we are talking about how to make this approach more feasible and more easier, then we have to talk how we can knock down CCR5 completely, like in Timothy's case.
Absolutely. Can Timothy be reinfected with HIV? He says he has this deletion. Can he be reinfected?
Yeah, like everyone who has this natural occurring mutation, there's a small but existent possibility to get reinfected by a mutated virus which doesn't require the CCR5 receptor. Yeah.
But the probability is low.
The probability is low.
In that case, okay. Timothy, so physically your life has changed, obviously, before and after. You lived for 10 years as an HIV positive man in an era that was not too far away from when the epidemic was in its height.
Right, exactly.
In the United States. So I'm coming from the place of the stigma that exists for many who are living with the virus, around specifically, having to disclose and confronting the whole notion of, would you want me? Would you want me friend? Would you want me partner, sexual partner, romantic, or what have you? How can I tell parents or loved ones, would you want me? So you live in that space for a while. Can you talk to us about how you handled that stigma of living with the virus at that time in your life?
The atmosphere in Berlin was much different than a lot of places in the United States. It's a very open city, and it's very accepting of gay people. And I told my workmates, I told my boss at work. I was working at a cafe on Checkpoint Charlie, and I didn't tell the customers, of course, but I told my workmates, and they were very open, very accepting. And I waited to tell my mother because she was dealing with her own health problems, and so I waited about a year to tell her. And I think I told my friend Elaine because I told her everything. She was the one that came and visited me in Berlin.
Right. Who came to say goodbye.
Yeah, right. I actually never really dealt with much stigma.
Being in Berlin.
Yeah.
With the virus.
And Yeah, so I understand stigma very well, but I haven't really experienced it that much myself. Coming out as gay in Seattle was hard, though.
Like in the 90s?
In the 80s.
Or late 80s, yeah.
Yeah, because I kind of came out in 83 and after, well, in my senior year of high school, and it wasn't very well taken.
Came out in 1983, senior year of high school.
Yeah.
That's huge.
And I was called a fag a lot, and I would walk down the hall, and people were like, "That fag." And
I felt that just now.
Right.
Yeah,
that hurt.
What I was impressed about with regard to your story, and those things are hurtful, speaking to that homophobia,
right,
that you kind of use that energy, and you got involved with activism. You were part of ACT UP, correct?
Yeah.
For a time.
In 89 and 90, Andy, who's sitting right there, and I we actually met at ACT UP, yeah, in the elevator, going up to in the elevator. And we became good friends after that, and he's still a good friend. And he is since married and has 2 kids, and yeah.
Wonderful.
Yeah.
Wonderful. Thank you so much for sharing, both of you, and this is only the first part. We want to give the crowd an opportunity to ask their questions, give their comment with regard to both your experiences through this amazing feat. What we're going to do now is we're going to ask, if you do have a question, to not raise your hand. I know, we're so used to being in school. But you can line up here on this side here, because what we're doing is because there's only one microphone, and that's me. And while I do like to exercise, I'm not going to run around the room this evening. But if you have a question, please line up here on this wall, and then I'll come to you, and you can ask your question to either Gero or Timothy. So feel free to come. And so this is it. This is how it goes. I hold the mic, though. I don't let go of it. I don't. I'll fight you for it. So, but feel free to ask whatever question you'd like. And if you don't mind, you don't have to, please state your name, but it's not obligatory.
Hello, my name is Sebastian, and I want to just talk to Timothy and Gero on, during, the time, Timothy, when you were, diagnosed with HIV, and Gero, when you were doing the procedure of the transplant. What was going through your minds?
Being diagnosed with HIV was hard, although the diagnosis with leukemia was harder. I am not a very emotional person except with movies. I cry during movies, and they always play they play the thing, so I cry. And I didn't really have much reaction to finding out I had leukemia. I was kind of shocked by it, and I realized that I not only had one death sentence, I had gone through that with HIV, but I had a death sentence, and a more direct death sentence with having leukemia. And go ahead.
Yeah. At the time point we made the transplantation, we knew that we are doing something very special, very interesting thing, which could change the whole medical world afterwards when it works. And this was the point we were not clear about what happened exactly when we do this. And so it was a bigger and good surprise that it works. So, yeah.
I was just going to add. Gero had told me he had presented a paper to the letter to the New England Journal of Medicine, and it was at first rejected. And I knew about the paper, and I kind of believed him, but I didn't really believe I was cured until they published the paper.
Hi, Gero, you're my hero. Thank you. And Timothy, you know I love you. So I'm my sister. I don't know, Tim, if I've ever told you this, but she went through a bone marrow transplant. I know it's brutal, and she had a rare blood disease. She's cured, but I attribute a lot of that to the care. She has a medical background. Her partner has a medical background, so she was there every day cleaning the tube, and there's a lot that goes with it in the aftercare. So I wondered if you could talk a little bit about that. And also, she's been in contact with her donor. I know this is a personal question, but I just wondered if you have been in contact with the donor since the transplant.
In Germany, you can contact your donor after 2 years, and the donor can contact you. And if both want to see the other person, then it happens. But in my case, I went and talked to Gero's boss, the Professor Thiel, and I didn't change the name to protect the not so innocent. And I told him that I wanted to meet the donor, and he said, the donor doesn't want to meet you. And I found out that that was not true from another source, and so it hasn't happened yet. It might. I have no idea if he knows that he caused such an uproar in the HIV community.
Interesting.
So as you probably know, right now, there's all this new cure research starting. It's all very exciting, but it's all very new, and it's all just kind of testing different things. And some of these things are potentially toxic or side effects and this, that, and the other. And then when you do these clinical trials, you know, things like leukapheresis and tissue sampling up the butt and all the rest of it, you know, and there's no potential benefit to the participant. It's purely altruistic. So this is kind of a bizarre question, but had your whole thing not happened, your other health issues that caused you to have the transplant and be cured and all that, would you have considered participating in one of those trials?
Possibly. I told Matt Sharp, who's here today, he gave a presentation. We both presented in Toronto, and for OHTN, which is the Ontario HIV Treatment Network, and he told about his story, and he has a fascinating story. I told him that he's my hero because I don't know where you are, Matt, but you're my hero because you actually chose to participate in these trials, and thank you for that. In my case, I didn't have a choice. It was do it or don't or die. So, yeah.
Thank you.
Okay. This is actually kind of a coincidence, and I wasn't planning on saying this, but on my way up here today, at my old junior high school, the current principal, I can't remember what kind of cancer it is, but they were having a drive to get people on the registry for with like blood cancers, and you can be a donor, and all they needed was like 4 cotton swabs, and it was also simple, and there's information about it. I don't know. It's Be the Match, and so it's bethematch.org. I don't know. I figured I'd say it. It's kind of a coincidence that it happened today because, like, my mom told me about it, and I was like, oh, hey, and then I drove by it, and sure enough, they were having the drive. So all they needed was just 4 cotton swabs, and then you're on the registry, and then, and then they might get a hold of you. bethematch.org. Anyway, this is my question. I was born in Tacoma, grew up around the area. You're in Seattle, and we just made it legal. Did you ever use marijuana-based medication for anything like stress or pain or anything like that?
I did not. I dabbled in junior high school and high school, and I didn't like what it did to my brain, and so I kind of quit. I didn't use any as medication. I'm now on the board of Cannabis Science, which is they do medical marijuana and research, and they're dealing with cannabinoids to reduce inflammation, which happens with HIV patients, whether you're on medication or not. And so it's very important. Another thing is that a friend of mine had a Carposi on his arm, and he used a cannabinoid oil and rubbed it on his arm, and the lesions fell off. So yeah, it's very powerful. And
CBD stuff? It's like CBD for
like that.
For when you don't want the cerebral effect or something.
Yeah.
Huh. Cool. Thank you.
Thank you. Absolutely.
Thank you so much. Timothy, I'm called Moses Supercharger, representing the community of Africa on the AIDS, on the ACTG and Insight clinical trials.
Thank you for that. Thank you.
You are the symbol of hope in this struggle, Timothy. I have 2 questions. One, goes to the doctor. Was there some kind of consultation from these people who have tried to do what you did to Timothy, like the Boston patients? Did they try to consult you so that you can maybe... Is there a possibility that there's something they can't do that you do? And my last question, I come from a continent whereby we have 70% of people living with HIV come from Africa. Hopeless. No hope at all. And these days, they hear so much about Timothy Brown. And I'm one of the luckiest set person that I can come to such an amazing meeting and see you. And normally when I go back to Africa, one of them actually asked me that, is there a possibility of getting those drugs that cured Timothy? It's really complicated in our continent. My question is there a possibility of bringing Timothy to Africa so that he can interact with millions of people living with HIV? Thank you.
I definitely want to do that, and I'm actually talking with my business partner, Dave Purdy, to do that, and also with somebody that brought me actually to Dave Purdy after an incident in Marseille, France, and I ran out of money and asked for help, and this gentleman helped me, and so they're actually talking about going to Africa and Uganda and Tanzania, and I can't remember. And anyway, so yeah, I definitely want to go to Africa, and I want to help there. Thank you.
Hi, I'm Lori. And I have a couple of medical questions. So one is, I'm always confused when they talk about the Boston patients as if it were very similar to what you did, because they didn't have the CCR5 deletion, and it seems to me that that's one of the key elements. And people are talking about the Boston patients as if that's a failure to replicate what happened with you, and that seems very different. So, and I'm wondering what your thoughts are on the necessity of the double CCR5 deletion. And also, I know we're looking at the possibility of doing autologous transplants so that you don't have to worry about the match, and I would imagine there'd be less chance of rejection. But then there's concern about what role did that whole, I always forget if it's host versus graft or graft versus host disease, that happens during the transplant from somebody else. So I'm wondering if the doctor could comment on that.
It's an important point. And I was wondering when they published or make that Boston patient public that they claimed some kind of cure. I was very astonished about this because there are reports from 30 or 40 patients who received allogeneic transplantation without this mutation, and they all relapsed from HIV when they take off the medication. So the data are quite clear, but there are some similarities between the Boston patients and Timothy. It's the try to find the virus in those patients. And in both in Boston, like in Timothy, we try to find virus with the highest detection methods with single copy assays. They made apheresis to collect a huge number of white blood cells. They saw that the Boston patients, like Timothy, had a decline of antibodies, so they're getting HIV negative. They are not negative, but they're getting an HIV negative. And they did many things what we did in Timothy too, and they were all similar to Timothy negative. But all these tests were done under medication, and in this moment they stopped the medication, one a couple of months, the other nearly 1 year after the stop of the treatment, the virus came back. So my understanding is that the ultimate test is if you are curing somebody, you have to take away the antiretroviral therapy and have to wait if the virus come back. Every other method is not sufficient to claim cure. So we were in Tim's case, we were very careful in using the cure word. In the New England report we published 1 year after the transplantation, we didn't mention this. And Jay Levy wrote an editorial for this, and he titled "Not a Cure, but Encouraging New Directions" or something like that. We didn't say cure. I don't know why he comes to cure, but that's obvious.
I went to a meeting in San Francisco with another gentleman that's here, Stephen LeBlanc, and he went up to do something like this, talk to the panel, and Rowena Johnson was there. She's also here, and the thing was titled "Cure a 4-letter word," and he asked me if he could introduce me, and so I stood up and turned red, bright red, and yeah. So that was kind of my introduction to public speaking, but I didn't speak at all.
Could you also speak to whether you think there's a necessity for there to be the host graft disease, or whether you think with an autologous transplant you would be able to have some success?
Yeah, probably you need two things for the success. One is the withdrawal of the CCR5, and the second is that you reduce the reservoir cells, the potential infective cells, to a minimum, so that if they are spreading, if they are replicating, and they have no target, the mutation, that the infection comes to an end, and they have not the chance to get mutated by replication. And if you have target cells where they can spread, you have the possibility that this whole procedure doesn't work. So probably you need both the CCR5 withdrawal and some kind of immune suppression what limits the number of infected cells. And this is the easiest way to do this is allogeneic transplantation, but allogeneic transplantation is not what you are going with many people. So probably you have to put this this technique into other medications, like you're combining some kind of antivirus gene therapy with other kinds of immune suppression. That are things that were now still going with the Sangamo trial, but have no results not yet.
Thank you.
Hi, I was wondering what is about the CCR5 delta mutation that protects against HIV? Like, what is it about the mutation?
The mutation had nothing to do with HIV. The mutation is much more older. It's probably several 100,000 years old. And according to this information, it's astonishing that 1% of the people have this mutation. So there must be a selection benefit for the carriers of this mutation, but no one knows exactly what this benefit is, but the mutation is much more older than HIV, so it had nothing to do with HIV.
And Gero, do we know where these mutations come from? There's different theories out there,
yes.
Bubonic plague, etc., those who survived it.
Yeah.
What do we know about where this mutation
We know that there's a north-south distribution, that we have the highest amount in Northern Europe around the Baltic Sea. And it goes down with a frequency to the south, and it's absent in Asia, in Africa, and in Indians. So it's a European mutation. So.
But Natalia Holt, who wrote CURED, told me that there are other mutations in Africa, so that aren't
different.
CCR,
They're different
CCR5.
mutations.
So,
but also these have nothing to do with HIV. That's accidentally.
Yeah.
So Gero, just to clarify, what is it about the mutation that does not allow or inhibits the virus from entering into the cell?
It's the mutation which cuts off 32 base pairs, and the gene product, the receptor, is not cannot enter the surface of the cell. It's not expressed. It's kept inside the cell, and HIV requires CD4 as one receptor in combination with CCR5. Only this works for cell entry. And if there are no CCR5 receptor, then it doesn't work. CD4 is not possible to change. Everyone needs CD4. You cannot live without CD4, but CCR5 is redundant, and you can live without CCR5. So this is a trick.
So it's like an open door on the cell, and without that door, HIV is not able to enter at all.
I had a question for the Dr. Gero. How difficult was it back then in 2005 to search for this CCR5 delta 32 mutation? I know today medical technology advances have made it fairly easy, but how did you go about finding that donor?
The technique is very easy. It's a simple PCR assay, and it is not very cost intensive. But you have to convince the registries who are having the data of the donors that they are contacting the donors and giving you blood samples that you can test. That was the main limitation, and it took a little bit time, but after that it worked very well. And now the registry which I worked with, they are testing the donors at the moment they get registered. So we have a very, very rapid access of this data.
German medicine.
If we have now one patient like him, it's a similar data request from the data bank, and we have the information into this in seconds. So.
Wow.
Hi, I'm Madison. I was wondering, how has your lifestyle changed since you found out you're HIV free?
So I was busy kind of recovering for over a year, and I was asked to go to an international AIDS conference in Vienna, and it would have been hop, skip, and a jump from Berlin to go there, and I actually had to say no because my ex-partner didn't want me to, and so I was kind of not in the greatest health, and then I suddenly decided that I needed to tell my story so that other people would have a chance to become cured as well, and so I decided I was going to be an activist toward a cure for HIV, and thank you.
Absolutely.
And that's what I'm doing, and beyond that, I don't really work. Yeah. But I go to the gym. I stay in shape, so.
Someone said that's work.
Yeah, it is.
Thank you both so much for coming today. So my question is for Dr. Gero. Since your treatment of Timothy has had such a huge impact on the HIV world, I guess, what has your role in the HIV community been since your treatment?
I think it's not uncommon that people who are not in the main group of HIV researcher have good ideas. And for example, Robert Gallo is a hematologist like me. And no one talked about violence at this point. And but anyway, I think I have connection with many groups in the world who are at this topic with HIV cure and stem cell transplantation and gene therapy, and but they're dealing for their own. I'm not a binding factor of them. I share my information and my knowledge, and I hope that someone and develops something what is a better way for curing more people.
Great.
Timothy, has your perspectives on prevention, HIV prevention, changed now having been cured, or how have they changed, or how do you look at prevention now on this side of HIV being cured?
I had a discussion about PrEP with pre-exposure prophylaxis with a scientist, a medical scientist in Marseille. And at that point, I kind of said, well, I don't know about PrEP. I think that we need to save the medication for people who have HIV, and they need to be treated first. But since then, I've changed my mind, and I think that PrEP is actually a new vaccine. So, yeah.
And for those that may not know, thank you. For those that may not know, PrEP is the pill Truvada, which is a drug that those living with HIV do take. It's a part of an antiretroviral therapy combination. What we've learned as of 2012 is that if this pill is taken daily, as prescribed, consistently by HIV-negative persons, that it can inhibit the acquisition or transmission, rather, of HIV of over 95%. Again, taken consistently daily, but it's one of the new advents in prevention right now, and so a very powerful, powerful drug.
Thank you.
My name is Alex, and my question is for both of you, kind of individually. I was wondering if you could both speak to the reaction that you received, both from the HIV community, from the medical community, and just kind of the world at large, and if you describe it as underwhelming or overwhelming, or how you both experienced it.
The reaction I've gotten has been largely positive, except the article that I wrote for Positively Aware was published in by Yahoo, and there I got really horrible comments. I thought they were funny. I laughed. I'm like, okay, whatever. And so I didn't take them seriously. But otherwise, I've gotten really great comments from people. I went to the International AIDS Conference in 2012, and I actually wasn't a guest, but I was there. And I had people like bowing down to me, and yeah. And I'm like, I'm not a god. And so, yeah, that was kind of funny. Yeah, generally very positive. I actually, a friend who, one of the people that had called me fag at school, I told him my story, and he's like, thought it was really cool. And yeah, my mother's friends seem to be very positive about it, and most of them are conservative Christians, so that's very positive. And also, I went for World AIDS Day 2012. I went to Saddleback Church in Southern California, in Orange County, and the Rick Warren is been kind of demonetized by a lot of people in the HIV community, but his wife has been doing work in Rwanda, and they've actually adopted lots of orphans of HIV patients from Rwanda. So they're doing tremendous work, and they were very, very kind, the warmest people in the world. And so, yeah, that was very nice. So yeah, I've had a very positive experience.
My problem was that after the case came public, and I received nearly every day emails or requests from people with HIV will say, please doctor, do the same for me. I give you any money, and please do this the same for me. And this is very hard to answer this. I try to answer everyone. But sometimes it was my main work at day to answer these questions. And yeah, thank God that the requests declined now. But I have still some. But this is the reaction of the community, and the other reaction from the medical or from the scientific side is that I have no very good feedback from my German colleagues. I have very good reactions from the US, and of course from my friends from Holland sitting there. And but that doesn't matter. I think this HIV research is an international thing, and so it's okay.
Gero, with regard to finding cures, plural, there are different different approaches that are being taken with regard to finding cures for HIV. Can you tell us, what are your thoughts with regard to the approach that you think we'll see kind of first on the scene scientifically with regard to progression? One, maybe you want to answer this question first. Can you kind of explain the different types of cures that there are? Because there isn't one kind of cure. Can you kind of explain that? I mean, we have Timothy Ray Brown's situation, and then we have the situation with the child that was called the Mississippi baby, which is a different type of cure.
Yeah, it's a controversy about how to define cure. And cure is a simple word, but in terms of HIV, with the retrovirus, which became part of your own body, it's hard to define cure. And for me as an oncologist, we will make it very simple. If someone survives 5 years his cancer, we call him cured. So that's the easy way to do this. But there's a discussion. If you are claiming cure, then some people accept only a sterilizing cure. That means that you won't find any trace of the virus in the body. And this debate has blocked a little bit the progress in this scene. And I think most of them now agree that the sterilizing cure is not desirable, probably, and not possible. And if you take this Boston patients, for example, it's not measurable.
Right.
And you won't be able to cut him into pieces and to look in every single cell.
And tell my autopsy.
Yeah, I think Timothy has something against this.
Why? Well, I've already had pieces.
Where's your pioneer spirit?
I've had brain biopsies, lymph node biopsies. Colonoscopies, gut biopsies, you name it, and I was told during the International AIDS Conference by a scientist from Utrecht, Utrecht, from the Netherlands. He said, "I know you intimately," and I looked at him like, huh? And then he said, "I have 7,000 copies of your blood in my freezer."
That's a bit awkward.
Yeah.
Nice to meet you.
To Kenya. And so the common thing is that we are talking now about functional cure. So we ignore that there might be traces of virus left, but we are talking about someone who has no medication, who has no HIV symptoms, and who will never get AIDS. So this is a functional cure. And it doesn't matter.
Is controlled in the body. The body found a way to control the virus.
And he's not infected.
So, I'm sorry?
He's not infected.
They're, for all intents and purposes, not infected.
So, hi, my name is Amandin from Remna High School. So I have two questions. So one of the questions are, so why did the HIV cure work on the Berlin patient the way it did? Like, what were the main reasons that it worked on him? Or were there like any specific reasons it did?
Was there anything, is your question, was there anything unique about Timothy that allowed this procedure to work?
No, that's he was at the right time, at the right place. So.
And I had a strong desire to survive.
And his mother would say, attribute to the power of prayer.
Right.
So my second question is, now that Mr. Brown has like Kung Fu stem cells, can you like take those stem cells from Mr. Brown and put it like into someone else and cure HIV?
It could be possible, yes.
That would be possible,
but
yes.
would they do it? Since I had cancer, would they do it at all?
No. That's the exclusion criteria.
They're done working on you.
True.
But as I figured out, that the probability that the rest of this tissue matching is very low. It's just 1 to 200,000 or something like that. So it's better to go in the registry and look on this.
Hi. I had a question, actually, because, I've been waiting to talk to you for so long and haven't ever really gotten a chance yet. One of my questions about CURE is, we know that there are disparities nationally here in our country and in other countries, but then global disparities. And I'm just wondering what both of your opinions are on what we could do to already create a fertile ground, so if a cure comes, we can make sure that it gets to places like Africa and Asia, where there might not be as much infrastructure even now for treatment and prevention. Is there anything here that the people here might be able to do to see that come to be before we actually have a cure?
Can I say something? The National Institutes of Health will not fund any project for a cure, an HIV cure, unless it can be taken to the developing world. And so the scientists have to prove that it can be used on mobile units in Africa and wherever, in Southeast Asia, wherever. And so that is definitely looked at. Go ahead, sorry.
Nothing to add.
Okay.
Hello, everyone.
Hi.
My name is Dider Abdullahi, and I'm from the University of Washington. So I have kind of a twofold question.
Okay.
So this first question relates, so what AZT, I believe, the drug, and what many drugs in the past for using HIV have done, is they've been targeting one of the enzymes called reverse transcriptase, which is an enzyme that HIV uses to code DNA in the opposite direction, which causes a lot of mutations and errors, which makes it almost impossible to cure HIV, because the progeny from one HIV will be completely different from its parents. So it's really hard to target it. So that's why the CCR5 receptor is really interesting. But my question is it possible to use, so this is a twofold. My first question, is it possible to use gene therapy using a certain type of virus? I know they were talking about using adenoviruses last week to encode a certain mutation, because every receptor is uptaken and destroyed and then upregulated throughout the body. That's just the cycle. So incorporating in the genome of normal individuals, instead of going through the dangerous process of stem cell transportation, just using using gene therapy of incorporating a segment of DNA or causing a mutation or inducing a mutation on the CCR5 gene segment that can make it non-functional. Has that been used?
Yes, both. The classical gene therapy with antisense or short hairpin vectors, this is already used, and what you said that you are mimicking the CCR5 delta 32 deletion is also used with using the zinc fingers. And both work. In both cases, you can reduce the CCR5 receptor, but in both cases, you're not able to block completely the CCR5 expression. So this is some major limitation up to date.
One of my main questions about what was going on with Sangamo is that they were saying that they use a virus to snip out the CCR5, CCR5. To create the delta 32 deletion. And I was worried that that might lead to other health risks for the patient. So I don't know. I know he mentioned Jay Levy. His lab was doing work on trying to do that without using a virus to get in. But he wasn't funded, at first he wasn't funded at all by the NIH because of that reason that I mentioned earlier. So yeah, I think it's possible, but I don't think that other risks should take place to the patient.
Okay, the second question is a twofold question too. So the first question is, I was reading your paper, the research poster that you give us, really interesting, and thank you so much, it's brilliant. My question was about, you mentioned how the CCR5 receptor is a G protein receptor, therefore it has a signaling cascade. So I'm assuming there probably have been drugs that have interfered with that signaling cascade that people have used to block that transmission, to inhibit the HIV virus from incorporating its genome. Has that actually been done, and has it been as successful?
Yeah, there are also different possibilities. You can block the CCR5 physically with a molecule called marrow, and that works, but it works not very well. And they're using another kind of gene therapy with a small peptide which binds together with the CCR5 and which inhibits the folding of the membrane and incorporation of the virus. So it's all, everything what one can be done is done. So, but all these attempts are working principally, but not enough to make a complete depletion of the receptor.
Thank you so much.
Yeah.
And I just want to say thank you for the question. Absolutely. You know, when I first met Gero, I just want to say a side note here. I was amazed. He looked so young, younger than me. And
He's younger than me.
I feel I'm pretty young, but I mean, I just young and you look good.
Yeah.
And so, you know, you hear someone who's participating in such a great feat. My mind, I think, subconsciously kind of went to, or maybe stereotypically went to, you know, this person, I don't know, he was 70-something years old. And 70 is the new 40? So it's okay. But I was just real, I was like, wow, this guy's like, this is good. You look good, Gero.
Hi, my name is Andy, and I have a question that may be a little bit more policy-oriented, but I think you can probably give some feedback. You mentioned exclusion in terms of people who can't participate. Let's say that there was research that indicated that this transplant of tissue could work as a cure. Unfortunately, or fortunately, there would probably be millions of people, including myself, who are of Northern European descent, who would be 1 in 150 chance of carrying this mutation, who can't donate because they have to either be celibate for 5 years if they have sex with men. So would we not have to change the policy of the CDC saying that you can't donate tissue or organs for 5 years if you go for 5 years without sex? Or not at all, if you're a man who has sex with a man?
I'm not sure how to answer that. I thought I heard the other day that policy is no longer in.
The change in policy was about blood donation, and it was lowered to 1 year of celibacy.
Okay.
I know I donated blood in Houston to get money to live. And I probably shouldn't have, because I was told later that if you were an MSN person, that you should not do that. But I did it anyway. Well, I didn't know the policy yet, but yeah. So, yeah, that should be changed. I agree.
Thank you.
Okay.
And just to let you know, folks, that, and it's probably apparent, obviously, but there cure research is taking place in the United States. NIH-funded cure research in 3 locations. One, Seattle at Fred Hutch, which is changed to Fred Hutch, not Fred Hutchinson Cancer Research Center, because Fred Hutch is a place for cures. It's now no longer just focusing on cancer, but other infectious disease, i.e. HIV, also in North Carolina and San Francisco. And so we are closer today than we were 5 years ago. When we will have a cure, we don't know. Gero, what do you say when folks ask, when are we going to have a cure?
Yeah, it's not easy to answer, but sometimes it just needs a little piece in this maze to make it work. And we're not. Our current knowledge doesn't allow to predict how we have a breakthrough, but it can happen every time. So.
And just thank you for saying that. And there's a lot of things out in the web, in that world there, that's suggesting that you can take different herbs and things of that nature to cure HIV. Scientifically, can you kind of set the record straight on any other cure other than the one that's happened with Timothy Ray Brown?
Yeah, that's right. Yeah, yeah. Well, we're working in this field to repeat this, to get more information from other patients, and I think one cure is not enough, as you always say. And I think if we are doing this and increasing the number of people who have successful with treatment, this would increase the pressure for other research groups to find more easier, feasible ways in mimicking and copying this approach.
And currently, there's a lot of false claims of people claiming that they can cure, but Timothy Brown is the only one that has been cured scientifically, and this is what we endorse. Well, thank you so much for your time, your talking about your experience. Are there any other closing words or comments that you'd like to leave with us before we begin to wrap up?
I was just going to say, holistic cures don't work. If you do them, you're going to die. And if you use them as your only source of preventing HIV and getting rid of it, you're going to die. You're not going to make it. And I had a good friend in Berlin who went to a holistic woman, and she had him. She took him off his HIV treatment and put him on herbs and stuff like that, and he passed away. Yeah. And that was very sad.
Antiretroviral therapy. Yes, Gero, would you?
Yeah, no, I'm looking optimistic and HIV research because we have done so many things, and most of them, you say I'm looking younger than I am, but I'm 45, so I was really scared from HIV when it came up in the 80s, and it was my time when my sexual activity started. So I was very scared about this thing. And at this time, and when I started medical school at 1992, there was no active treatment, and I saw the people dying in front of me, younger than me, and. So this is what I kept in mind from HIV, and we are now much more far and as a chronic disease, and we have still unsolved problem like in the developing countries in Africa. There's still an uncontrolled disease. So we have many things to do still, but I think we are much more advanced than 30 years ago, and so there's hope for me that we are looking another, not 30 years, but 10 years. We have much more advance in this direction.
Absolutely.
And I always say that I think that there will be a cure before I die. So, so nobody kill me.
No, that's not. If you're thinking about it, stop. Absolutely. Thank you. And of course, around this, I mean, what's better than being cured is never having to be cured. And so testing, make sure you're tested. Yes. And if you learn that you have acquired the virus, see a physician. And we encourage begin antiretroviral therapy as soon as possible. Protect yourself and those around you. Consider other preventative modalities. Obviously, we know there's condoms, but there's also PrEP, which we spoke briefly about. And so consider the safety of not only yourself, but those that are in your community as well. Thank you so much for your time. Let's give both of these men a round of applause.